Say your last name out loud, just once, in whatever language you carry it. If your name is Cohen, or Katz, or Kagan, or Khan, or any of the 67 variations of that root word recorded across 30 languages, something extraordinary may have just happened that you don't know about yet, because that name is not merely a label your great-great-grandfather carried off a ship somewhere. Researchers now believe it is a biological signal, a marker embedded in a specific stretch of your Y chromosome that has been passed father to son for somewhere between 3,000 and 4,000 years, long before surnames existed, long before the countries your ancestors came from had names, long before anyone on Earth knew what a chromosome was. The name Cohen, in Hebrew, means priest. It is the designation for the ancient Jewish priestly class, the Kohanim, who, according to both Jewish tradition and the Hebrew scriptures, descend directly from a single man, Aaron, the brother of Moses. That is a lineage claim stretching back to the 13th century BCE. It is, by any reasonable standard, an unprovable founding myth, except that in 1997, a team of geneticists in Israel, Canada, and the United Kingdom ran a study, and what they found has not stopped reverberating through the scientific literature since. For most of the 20th century, historians and anthropologists treated the Cohen identity as exactly what it appeared to be, a religious and genealogical tradition maintained through community memory, oral transmission, and rabbinical record-keeping, not biology. This was a defensible position, a reasonable position. Here is why the consensus held. First, the Jewish people have experienced extraordinary diaspora. By the Common Era, Jewish communities existed across Egypt, Persia, Rome, Greece, and into the Arabian Peninsula. Centuries of separation, forced conversion, migration, and persecution had fractured these communities into distinct cultural groups, speaking different languages and following different local customs. Genetic continuity across that kind of dispersal for over 2,000 years was considered implausible. Populations mix. Lineages blur. Second, the Cohen designation itself was transmitted socially, not biologically verifiable. Fathers told sons. Communities kept lists. Rabbinical courts adjudicated disputed claims, but there was no external check. Anyone with a credible family story could carry the name. The designation, in other words, looked exactly like what most inherited surnames look like, an identity assigned and maintained by human beings, not written into the body. Third, the sample sizes and technical limitations of early population genetics made diaspora-scale studies nearly impossible before the 1990s. Even researchers who suspected there might be a genetic signal in the Cohen population had no reliable way to detect one across geographically separated communities. The science simply did not exist at the scale needed. The assumption was not careless. It was reasonable, and it was wrong. The anomaly begins with geography. Cohen men are not clustered in a single country. They are dispersed across North America, South America, Europe, the Middle East, North Africa, and the former Soviet states, communities that have been geographically isolated from each other for anywhere from 500 to 2,000 years. Many of these communities did not maintain contact with each other. Some did not even know the others existed. And yet, when researchers began collecting DNA from Cohen men across these different populations, Ashkenazi Jews from Eastern Europe, Sephardic Jews from the Mediterranean and North Africa, Mizrahi Jews from Iraq and Iran, they found something that a purely social identity system cannot explain. Cohen men from communities separated by 1,000 years of isolation shared at unusually high frequency a specific pattern on their Y chromosome. That pattern should not exist if the Cohen identity was entirely a social construct. It should have dissolved into the background noise of normal population genetics. Surnames drift. Communities intermarry. People adopt names for political and social reasons. Over 2,000 years, a purely social label should have become genetically indistinguishable from the non-Cohen Jewish population around it. It had not. Before going further, it is worth being precise about why this went undetected for so long, and why the credit for finding it belongs not to accident but to a very specific intersection of three technologies arriving at the same historical moment. For most of recorded history, genetics did not exist as a discipline. For most of the 20th century, the techniques available to population geneticists, blood type analysis, protein markers, early restriction fragment analysis, were too coarse to detect fine-grained Y chromosome patterns across globally dispersed populations. The Y chromosome itself was, until the late 1980s and early 1990s, considered relatively uninformative for genealogical purposes precisely because it accumulates mutations slowly. The research communities that might have asked this question, Jewish historians, biblical archaeologists, population geneticists, were also largely not in conversation with each other. The question of whether a religious lineage claim might have a detectable biological signature sat in a gap between disciplines. Historians considered it outside their method. Geneticists considered it a social science question. Neither was wrong, given the tools available. What changed was the convergence. High-resolution Y chromosome typing became possible. International sample collections became feasible, and a team of researchers was willing to sit at the intersection of all three disciplines at once. In 1997, a research team led by Carl Skorecki at the Rambam Medical Center in Israel, working in collaboration with Michael Hammer at the University of Arizona and Neil Bradman at University College London, published a paper in Nature titled Y-Chromosomes of Jewish Priests. The study appeared in Nature, volume 385 on January 2, 1997. The technique they used is called Y-chromosome haplotype analysis. Specifically, they examined a set of genetic markers on the non-recombining portion of the Y chromosome. Here is what makes the Y chromosome unusual and why it matters for this question. Unlike the rest of the human genome, which shuffles itself during reproduction, the Y chromosome is passed from father to son almost entirely unchanged. It is the most direct biological record of patrilineal descent that exists. Think of it like a wax seal that a family stamps on every letter they send. Not the content of the letter, which changes, but the seal itself, which is copied identically each generation. The non-recombining portion of the Y chromosome accumulates mutations called single nucleotide polymorphisms, or SNPs, at a known and relatively stable rate. By mapping the pattern of these SNPs, researchers can cluster men into groups, haplogroups, that share a common paternal ancestor. They can also identify specific short tandem repeat sequences, known as STRs, that create a finer-grained signature called a haplotype, a kind of molecular fingerprint for a particular patrilineal lineage. Skorecki, Hammer, and Bradman collected DNA from 188 Jewish men across Israel, the United Kingdom, and North America, both Cohen men and non-Cohen Jewish men. They selected participants carefully, men who could demonstrate through family testimony a consistent Cohen or non-Cohen status without mixed or disputed claims. What they found was this: a specific Y chromosome haplotype, subsequently named the Cohen Modal Haplotype, or CMH, appeared in approximately 46% of Ashkenazi Kohanim and approximately 56% of Sephardic Kohanim. In the non-Cohen Jewish male population, the same haplotype appeared in roughly 3% to 5% of men. To be precise, the same molecular signature, appearing at roughly 10 times the background rate, in two populations that had been geographically isolated from each other for over 1,000 years. The haplotype is real. It is old, and it crosses every boundary that should have erased it. Shows. The 1997 Skorecki et al. Study showed that Y chromosome haplotype frequency is significantly elevated in men who carry the Cohen designation, regardless of whether they belong to the Ashkenazi or Sephardic branch. The finding was replicated and extended in a 2000 follow-up study by Hammer et al., published in PNAS, Proceedings of the National Academy of Sciences, which analyzed a larger sample across 6 Jewish and 3 non-Jewish Middle Eastern populations. The CMH was found to cluster most closely with Y chromosome patterns in contemporary Middle Eastern populations, consistent with a geographic origin in the ancient Levant. A 2009 analysis, using higher resolution markers by Behar et al., published in Nature, further confirmed the distinct Y chromosome profile of the Kohanim. The data rules out several alternative explanations. It rules out random convergence, the probability of the same haplotype appearing at this frequency in two isolated populations by chance is, according to the researchers' modeling, extremely low. It rules out social adoption as a primary mechanism if the Cohen identity had been widely adopted by unrelated men over 2,000 years of diaspora. The Y chromosome signal would have been diluted to statistically insignificant levels. It rules out regional genetic drift as the cause. The signal appears consistently across populations from Eastern Europe, the Mediterranean, North Africa, and the Middle East, geographically too dispersed for local drift to explain the pattern. And it does not rule out, but does substantially complicate any simple claim that the CMH proves direct descent from a single historical individual. Implies, here is where researchers are careful and where this video will be equally careful. The data implies that a significant subset of men carrying the Cohen designation share a common patrilineal ancestor who lived by mutation rate modeling somewhere between 2100 and 3900 years ago. A range consistent with the biblical dating of Aaron, but also consistent with a priestly clan that consolidated power several centuries later during the First or Second Temple period. The data does not confirm that this ancestor was Aaron specifically. It implies that paternal line transmission within the Kohanim has been maintained with unusual fidelity across the diaspora, something that population geneticists call strong patrilineal selection, driven, the authors suggest, by the social, religious, and legal consequences attached to the Cohen designation in Jewish law. The title conferred obligation and restriction. That friction may itself be what preserved the lineage. There are a few things I haven't told you yet. One is what happens when you apply higher resolution analysis to the CMH and what it reveals about the specific branch of that lineage most Kohanim belong to. Another is what the data says about the women in the story. The mothers, whose mitochondrial DNA tells a very different and in some ways more complicated history. If that interests you, subscribe. Everything published here follows the same method: primary sources, named researchers, honest limitations. I want to be honest about where this gets complicated, because the CMH finding has attracted serious criticism, and not all of it has been dismissed by the field. Counterargument 1 comes from geneticist Sergio Della Pergola and others who have noted that the CMH appears in some non-Jewish populations at rates that complicate a simple origin story. A version of the haplotype has been detected in some non-Jewish populations in the Middle East, which suggests either that the haplotype predates the emergence of Judaism as a distinct tradition, or that there has been more gene flow between Jewish and non-Jewish populations than the founding myth implies. The researchers do not dispute this, they explicitly acknowledge it. Counterargument 2 is more fundamental and comes from within genetics itself. The CMH, as originally defined in 1997, used a relatively small number of markers. Subsequent high-resolution analysis by David Goldstein's group at University College London, published in 2001 in the American Journal of Human Genetics, found that when more markers were analyzed, the picture became less clean. Not wrong, but less dramatically convergent than the initial study implied. The CMH is real. Its age range is genuinely uncertain. Hondi. I want to be honest about where this gets complicated in my own reading of this material. The temptation, when you encounter a finding this striking, is to round up, to make the finding more definitive than the paper actually claims. The 1997 paper is careful. The follow-up studies are careful. The popular coverage of those studies, including, I suspect, some coverage you may have seen before finding this video, frequently is not. There are today an estimated 8 to 10 million Jewish people worldwide who identify with and practice Judaism, along with many more who carry Jewish ancestry in various forms. Among them, the Kohanim represent a living, functioning community in Israel, in the United States, in France, in Argentina, in Australia. In traditional Jewish synagogues, Kohanim are called first to the Torah reading. They participate in the priestly blessing. In communities following halachic law, They observe specific restrictions around death and marriage that are still practiced today. The Cohen Modal Haplotype study was not conducted on a historical population that no longer exists. It was conducted on living men who carry a living tradition, embedded in living communities that have maintained this designation through persecution, expulsion, and cultural pressure for 2,000 years. Whatever the molecular story says about deep ancestry, the social reality of the Cohen identity is not reducible to it. The gene does not make the priest. The community does, and that community continues. There is something unusual about this particular finding in the history of genetics. Most ancient lineage studies describe loss, populations that disappeared, migrations that erased what came before, admixture events that dissolved an identity into something unrecognizable. This one describes the opposite, a patrilineal signal that survived 2,000 years of dispersal, forced migration, and cultural rupture with its frequency largely intact. Survival, in genetics, is rarely accidental. Something preserved this lineage, not instinct, not geography, but a social institution strong enough to constrain paternity patterns across 30 countries for 3 millennia. That is not a minor footnote in population genetics. That may be one of the most striking examples of cultural selection acting on a biological lineage in the modern human record. What remains open is a question worth holding. The CMH identifies a common ancestor, but there are other Kohanim lineages. Men who carried the priestly designation whose Y-chromosomes do not cluster with the modal haplotype. If ancient Levantine genomes from the First and Second Temple periods were ever recovered and sequenced with sufficient resolution, would we find multiple founding lines beneath the one we have named? Or would we find something stranger, a population bottleneck so severe that it reduced an entire priestly class to a handful of male ancestors within a specific century? That question has a specific shape. It points to a specific archive, the cemeteries, ossuary collections, and archaeologically recoverable deposits of ancient Judea. The answer, if it exists, is in the ground.